Perfect. Thank you so much for the introduction. I was looking at the participants as people came in, and there are lots of familiar names and a few familiar faces in here. So thanks for coming today, and thanks for that really nice introduction. I made slides for today. I'm going to talk through my path, my background, projects I've worked on, and hopefully one of the take-home messages you'll get is that like professors don't typically have a linear path. So you're going to see the path, even if one step didn't necessarily make sense leading to the next, but I'll try to explain my decisions and how I got there. I will keep an eye on the chat, and feel free to make this more of a discussion and jump in. And if something's not clear, definitely interrupt me. If we start taking too long, I can just speed through one part or not cover it. So I do have slides, so I'm going to share my screen now. Oh, Christina, do you mind enabling me to share screen by chance?
Yeah, I just did it.
Oh, perfect. Okay. Sharing my screen now. Okay. All right, so most students call me Professor Ellis. You can call me that. You can call me Shannon. I don't care either one. So I figured I would title this My Path to Data Science and My Path to Data Science Education. I always found it helpful when I was in school to learn more about like how people got to where they are and how they made those decisions, and just kind of learn those details. So hopefully y'all find it helpful. And so I'm starting back in high school and going on the path of how I got to here. So we're going over more than a decade here. And part of the inspiration, aside from Christina asking me to give this talk, was this tweet recently. Data Science Renee is a data scientist on Twitter, and she tweeted, let's show what a wide variety of career or educational backgrounds data scientists come from. Reply with your job and degree history. And she gave hers. And so here is mine. It's not a tweet, but there's a list of my jobs starting in my early teenage years, all the way up until my current position. In the chat, just from those jobs alone, does anybody know where I grew up? Feel free to. Somebody said the Midwest. Somebody said Southeast. Oh, this is so interesting. Okay, and so this is also borne out by the fact that we don't have that many students at UC San Diego from this part of the country. So I am from right outside Philadelphia. Rita's Water Ice and Wawa and Dave & Barry's are all like very Philly-centric companies. And so Wawa is the best if you ever find yourself on the East Coast. It's a place to go get the best hoagie. And so I'm from right outside Philadelphia. I grew up outside of West Philadelphia, and I had a job at a water ice store, which was awesome because as a teenager, I got to eat all the free water ice that I wanted. If you don't know what that is, it's like Italian ice and there was custard. It's really good. That's getting you through high school. I was a tutor. There's a comment in the chat. Anytime I say, yeah, West Philadelphia, born and raised on the playground is where I spent most of my days. Okay. I then in college, I was a D3 athlete. And so I had an irregular schedule, and tutoring worked out really, really well for me because I could make my own tutoring hours. And so I tutored one-on-one. I did mainly biochem, chemistry, and genetics. From there, it did become kind of research focused. So I did a research position. The ones in bold, I'm going to talk about a bit today. I did research at my home institution. I'll talk about what that was. And then I got a bachelor's in biology with my second major in Spanish, a PhD in human genetics. I'll talk about this path. During my PhD, I really like hanging out with kids. And so I wound up babysitting on the side, and I tried to not let people pay me money, but some of them insisted. So that was kind of a side hustle. My postdoc was in biostatistics. I started teaching. I taught at the Centers for Talented Youth. I taught at Hopkins. And now I'm here, which is how you all have gotten to know me. I arrived here in 2018. And so I kind of wanted to normalize that there is no normal path and that there's not one way to do this. I'm going to tell you about my path, but feel free to stop me anywhere in this and ask questions. But I'm going to kind of walk you through the four like main components. One was what I did as an undergraduate as a biology major. I noted here a lot of time with fungi and flies. In grad school, I was studying the genetic basis of autism, so like a big old pivot there. And then in my postdoc, I'll explain there was another big pivot. And then that led me up to my move to San Diego and teaching here in the cognitive science department. So these might all somehow fit together, but they do seem pretty disparate at the start. Okay, so just to like give you a sense, I went to a huge, it was amongst, and I think it's now the second or third largest high school in Pennsylvania. And I was a really good student. I liked school. I sucked at taking standardized tests from elementary school. I know people say that it feels like a cop-out, but I'm like pretty consistent in how horribly I do on standardized tests. So at that point, I was applying to some really good schools, but I didn't know what path was right for me. I liked a lot of different things. I liked writing, I liked history, I liked politics, and really, I had a really great biology instructor or teacher in high school. And in that, when I learned about Punnett squares, I thought genetics were awesome. And so I decided to go pursue biology, but I could see myself going in lots of different directions. And just as a note, like there is lots of rejection through this path. I'm going to try to highlight a bit of it. I applied to a bunch of really good schools and I got rejected from a lot of them, including Hopkins, where I ultimately went for my PhD, including what I thought was my dream school, which was Cornell. I hate the cold. I don't know what I was thinking. I would have been really cold there. And I got waitlisted at my safety school. So that's the one you're supposed to get into. I ultimately was deciding between the University of Pennsylvania and King's College. So in this, I'm going to try to highlight where I had to make decisions. And so I'll have this decision point slide and try to tell you how I ultimately made the decision. And there was a comment in the chat, overqualified for the safety school. I still don't know to this day. As it happened, I was really surprised. It's a good school, don't get me wrong, but I really felt like I was a good fit. And it's just one of those things, like sometimes you can think you're a good fit and there's just a lot of good applicants and they reject you. So I got waitlisted. I don't know why. There were jokes after it. They were like, you should send your University of Pennsylvania acceptance letter to them and let them know they made a mistake. But these things happen. And so ultimately, I was deciding between University of Pennsylvania, which is an Ivy League school, and King's College, which there is no reason any of you would have ever heard of it. It is a super small, I'm talking 2,400 students total, liberal arts college in Wilkes-Barre, Pennsylvania. If you've never heard of Wilkes-Barre, you may have heard of Scranton because of The Office, and Wilkes-Barre is near Scranton. Like that's how we get Wilkes-Barre on the map. And so I was between these two very different schools. And my parents are very good parents and would not help me make this decision. And so they were kind of like, it's up to you. So the reason I applied to King's College was because after I started panicking that I wasn't going to get into any colleges, I decided to apply to a rolling admission college. And that's what King's College was. For Penn, I really wanted to go there. I thought it'd be a great school. The reason here, reason I was deciding between these two is King's College offered me a full academic scholarship for 4 years. And so I wouldn't pay any tuition. And University of Pennsylvania gave me a nice financial aid package, but I still would have been paying like $22,000 a year. There's a question in the chat about rolling admission. And rolling admission, I don't know how much this applies to UCs, but on the East Coast, some schools, all the decisions come out like at one point. And if you don't get into any of them, you're not going to college. Whereas rolling admissions, they admit people on a rolling basis. So like as applicants come in, they'll send out admissions. So you can get accepted before that like drop dead deadline where you're just not going to go to college. Yeah, so there's a comment. There is a King's College in London. There's also one in New York. This is neither of those. King's College in London is quite a good school. So anyway, I was deciding ultimately between King's College, where I wouldn't pay any money to go and get my undergraduate education, and Penn, where I would be like $80,000 to $100,000 in debt coming out of it. And I'm nothing if not frugal. And so King's is really ultimately where I went to because of the money. After I made that decision, my mom told me she wished I had gone to Penn, and that was her dream school. And she couldn't afford it, couldn't go there. And so because I had the opportunity, she wanted me to. But I ultimately decided to go to King's College and study biology. Okay, so that brings me to this first bit of research. How did I start on the path that I'm on? And so I told you I tutored a lot as an undergraduate, which was awesome. I loved tutoring. But I also got involved with research pretty early on. And I'm going to try to highlight the mentors I had along the way because they really formed the like scientist I am, the instructor that I am. And so at King's College, Jeremiah Ori was my undergraduate advisor. And I did a summer, what's known as REU, so research experience for undergraduates at Carnegie Mellon in Pittsburgh. And I did that with Joe Ayub. And so he was my mentor there. Okay. So something that happens at small liberal arts colleges that would never happen at UC San Diego is I was taking genetics my freshman year of undergraduate, and I was mentioning to like my lab partner in lab, I guess, I don't even remember doing this, that I was interested in doing research going forward. So as I was coming into undergrad, I had this idea that I might want to do research, even though I had no idea what that was. And Ori, so I'm going to call him Ori. I should call him Jeremiah, probably would be his preference at this point. So Professor Ori, or Dr. Ori, as I knew him as an undergrad, he emailed me after he overheard this in lab with this quote that I've here. I heard you mention in lab that you're interested in doing research. Would you like to join my lab? And this is one of those like serendipitous, super lucky. At that point, he knew I was a reasonable student. Like I'd been in his class for a little while. And so he reached out, and that was how during my freshman year of undergrad, I started doing research. And that just like would not happen at UC San Diego. Okay, so what research did I do as an undergrad? I'll have to have you know that I like dug into the vault. I had to go find an old hard drive to get these images off of them because I have not thought about this work in years, but I did it. I found them. And so I pulled a few images to just kind of describe briefly what I did as an undergrad. So no details here. What you need to know is Cryptococcus neoformans, or C. neoformans, is what's known as a yeast-like fungus. So the pictures you're looking at are this fungus growing on a plate. So you could picture like a plate that's like this big with agar on it, and then these fungi grow on these plates. And the difference between what you see on the left and the right is that when copper, which is what copper sulfite is up at the top left, is in the plate, the Cryptococcus neoformans is healthy. So healthy is like these round, circular, can grow. You know, once you get up to 1 millimolar, which is this highest concentration at the bottom, that's when it starts to get a little sick. BCS is a chelator, I mean, it sucks copper out of the environment. And so there, while it can still grow, it's not quite as healthy. So what I want you to know from this is Cryptococcus neoformans needs copper to grow healthfully. The reason that matters is Cryptococcus neoformans is not a risk, and it's all over the environment to individuals who have fine immune systems. But if you are immunocompromised, coming into contact with Cryptococcus neoformans can be fatal. And so we set out to try to understand how to affect what's known as the virulence, how to make it not be able to grow inside a human host. And so copper is one of those virulence factors. There's other ones. There's this like cool sugar coating on it. And all of these help make it dangerous to humans. And we're trying to understand how we could stop it from being so dangerous to humans. And so don't worry too much about this very biology-centric image here. But what you need to know here is there are ways to control whether or not certain genes are turned on or turned off, and copper is part of that pathway in Cryptococcus neoformans. And there's a specific thing in the genome of Cryptococcus neoformans called CUF1P. And we were interested in how that affected the ability for Cryptococcus neoformans to be healthy. So in genetics speak, there is a triangle up at the top. The delta here means knockout. So we knocked this gene out of the Cryptococcus neoformans and saw how it grew. in when copper was around. So wild type on the left is normal, what you would find out in the wild. On the right is if we got rid of this, this gene, and we can see that we are able to knock down its ability to grow. And so the idea is, if we understand this pathway, is there a way we could target this inside the human body to stop it from being able to make people sick? If there's a question in the chat, will the diagram be on the quiz? There'll be a really strict quiz at the end of this, so you better memorize everything. Okay, and so the details on the right here, again, don't matter, but we were trying to characterize how this knockout strain differs in how it handles copper relative to what we found out in the wild. So all that's background. There's a question actually scared you, thought you actually had a quiz. I guess I shouldn't say that when most of y'all are actually used to having me in class where there are quizzes. I promise there's no quiz. And so this here is a slide that I presented as an undergraduate, not my best work, but I think it makes it an okay idea. So we had this wild type strand on the left, and we had it with copper treated. I would extract what's known as mRNA. Again, details don't matter. I would put these fluorophores on them. So one strain was green and one strain was red. And then I would hybridize them to these microarrays. These microarrays had every single gene in the Cryptococcus neoformans genome on it. And I would say, okay, between these two different strains, which ones differ between the two? And so that and those experiments and the analysis of those are really what I did as an undergraduate. Okay. Any questions through that whirlwind tour of Cryptococcus neoformans? All right. I'm gonna try to also put these quotes throughout that I think were really influential, even if I didn't know it at the time, in the path that I ultimately took. So as a background, I spent months, literal months, doing experiments at the bench to get these microarrays ready. When they were ready, we got this picture of this little slide at the bottom by putting it into a scanner, this very expensive scanner, and getting out these images of all these spots. And so then we had to say, okay, which spots are really green, which spots are really red, and then what's the relative ratio on all of the other spots? Because those were really the data we wanted to analyze. And so I spent literal months for every time I did an experiment of this type, getting the data. I then put them into the scanner, got my image out, and then to analyze the data, we used this software where it took the picture and then told me which genes were differentially expressed, which was really what we were looking for. So it took these green and red spots and all the colors in between and then said, okay, these are the ones you're most interested in. And I said to, at that time, Dr. Ori, I was like, whoa, what just happened in that software? Like, I spent months getting these data, and like, what did that software just do? And he said to me, if you weren't about to graduate, I'd teach you Python. And I like threw that in my brain because I didn't know what Python was. And nobody up to this point in my life had ever suggested that I learn to program. I wasn't a big gamer, and that was the only idea I had about programming. Like, I thought if you wrote code, you made video games, right? Like, that was my understanding. My high school, being so huge, had tons of like AP, those advanced placement courses, and it had an APCS course. Not a single soul ever suggested that was something I should look into. And so it was this moment where I went back to my dorm room and Googled what Python was, because I didn't know. And so that was the first like seed that was planted that might be something that would be helpful for me to learn. Okay. In the meantime, I'll do this one pretty quickly. This was one of the summers that I was in undergrad. Ori, my advisor, suggested I apply to these REU programs, these research experience for undergraduates. I applied to a bunch of them. I got rejected from lots of them. I got accepted at Carnegie Mellon in Pittsburgh. And I spent my summer making these videos. So what you're looking at here, and I'm going to play them. Again, these came off the vault, like deep into an old hard drive. On the left-hand side is a Drosophila melanogaster embryo. And so if you don't know what Drosophila melanogaster is, it is a fruit fly. Geneticists love studying fruit flies for unrelated details. One of our favorite model organisms. And what's on the left-hand side is the normal development. And what you're going to see here is at the head, which is off to the right, there are a bunch of cells forming. And over time, a few of them rescind back and you get this little loop that forms toward the head. So this is the anterior of the head of the fly as it goes through development. Before I got to the lab in the summer, there was some evidence that HSP27, which is this heat shock protein, led to what's known as increased apoptosis or cell death, and that too many cells were dying. And so for reasons that don't matter a ton, we were interested in understanding that more. So I spent my summer injecting fly embryos with HSP27 knockdowns. That's what siRNA stands for. And taking these, what I think are cool videos. So here's an example of one of these videos. And so we saw that rather than normal development, you get this extra space and all of this extra cell death. There's just a whole bunch of stuff dying at the front of this embryo. And so I made a whole bunch of these videos on a whole bunch of different fly lines, which means like different genetic variations of fly lines. And these were the results. So what you see on the x-axis, all the way to the left is wild type, right? If we didn't make any genetic alterations, how many of them had the mutant phenotype? And you could see 1.3% had this mutant phenotype with all the cell death and that extra space. And then all of the other bars are different lines. Again, this was a slide from my summer presentation as an undergraduate. And the green bars are those that were in this important part of this region of the gene. And we saw that not all of the flies, but a higher proportion of them relative to wild type had this phenotype. And so we were first time finding that HSP27 had something to do with this process of apoptosis and cell death and development. All right, that's whirlwind tour. We're now post undergrad. We're in the 2010s. Questions up to this point? I like that comment in the chat. How many people went into computer science to make video games? Yeah, I mean, I didn't go into it because I didn't want to do that. I'd imagine there's a handful the other way, especially when like programming the way it's talked about now as being such a helpful skill just was not the case, right? Like it was not discussed the way it is now. Okay. So as an undergrad, I studied this fungus and I did this summer researching flies and then applied to grad school. And so I'm going to comment on the chat first. I wonder how much gender stereotypes played into that being the first time programming was suggested to you. It's something I've thought about a lot. There's like, I'm sure that's part of it. I'm also sure like I had expressed interest in literature and history and biology. And so like maybe, you know, teachers didn't feel the need to push me in a different direction because I had enough interest, but I do wonder about that. Okay, so I'm in undergrad. I'm at a not very academically rigorous school. Y'all, I had a lot of fun in undergrad. I played 2 sports. I did well in school, but it was not the most academically rigorous or stimulating place. And so when I was transitioning to grad school, I applied to a lot of different programs, and I wasn't sure how well prepared I was going to be for my graduate school program. And so I also, again, there's a common theme. I like a lot of different things. So because I loved the research I was doing, I really enjoyed research. I applied to PhD programs. I applied to MD PhD programs, which is where I would be a physician and a PhD coming out of it, and also to PhD/genetic counselor programs across the country. Y'all, the like spring semester of my senior year, I was just like interviewing all over the country and flying somewhere every other weekend. It was a lot of fun. It was also exhausting. And ultimately, I only got into one MD PhD program. Another theme, my MCAT scores sucked. Those interviews are a discussion for another day. People were kind of jerks about how low my MCAT scores were on those interviews. I didn't get into the PhD/genetic counseling program at VCU, so Virginia Commonwealth University, but I did get into most of the PhD programs I applied to. And so I had to decide between ultimately Johns Hopkins for a PhD in human genetics or Virginia Commonwealth for a PhD in human genetics. And I struggled a lot with this decision, like enough that Hopkins was calling me every other day, being like, you know, you got to make a decision. And I was like, the decision date is whatever. I have until then to make the decision. And ultimately, I asked a lot of people for their advice. And the best piece of advice I got there was actually from Joe Ayub, the REU mentor who helped me with the flies. And he said, which program will you regret not going to, right? If everything else is equal and you're excited about both, you'd be willing to live in both cities, you're excited about the coursework. He's like, which one would you regret not going to? And that's where I decided to go to Johns Hopkins in Baltimore, Maryland for my PhD. I figured that would open as many doors as possible because I wasn't sure exactly what I wanted to do after my PhD. Okay. I'm loving the comments in the chat. For those who don't know, I love baking, and I joke that my backup career is to open a bakery and like go to culinary school. So all of these CSs, cognitive science, computer science, culinary science, these all fit in with who I am today. So this is a really great question in the chat. So I'm going to go back. How important would you rate prestige of grad school when applying for professorships? So there's a few. It matters more than it should. That's going to be my answer. It will open more doors if you go to a more prestigious school. I don't think that's how it should be. I know that my resume got looked at more as I was applying to jobs, including this one, because Hopkins was on my my diploma and because I was getting rec letters from people there. So it should not be the only thing you think about. And I wish it mattered less, because honestly, we could talk about, and we will talk about just in a second, the mentorship I got in my PhD was amazing. The coursework I did in my PhD, the instructors were not so great. And that's because this guy right here, Don Arking, my PhD advisor, amazing advisor to me as a research student. That man lectures 3 times a year, 3 days a year. He gives the same lecture once a year. When you're not teaching all the time, you wind up not being a great teacher. And Don knows how I feel about the lectures he gave. I told him every year, I was like, you need to improve them. I will improve them. And he was like, well, they went fine. Nobody asked any questions. And I was like, yeah, everybody was confused, Don. Can't ask questions when you don't know what's going on. So anyway, I didn't have the best instructors in the courses I took in grad school. So yeah, I will give you all the harsh truths here. So even though this is recorded and however shared, none of that is a surprise to Don. He and I talked about it all the time. Okay, so I try to point out my mentors here because they really did guide me along the way, give me a ton of knowledge, and help me in making all of these decisions. And Don is no different. So he was my PhD advisor. And so I joined the Arking Lab in my first year of graduate school. But I kind of wanted to point out that like it's not just your mentor, and especially in graduate school. So this is a slide from my thesis presentation. Again, pulling it out from the vault. And these are the people that day in and day out, like Don answered so many of my questions, but these are the people that like made graduate school enjoyable and answered all of my questions that I had that were, you know, what I'd asked before I went to Don. And so I'm like still very close friends with at least half of these people and talk to all of them regularly. And so it's not just my mentor, it's the people that I worked through every day. Okay. So I'll pause there. Any discussion points or general questions people have up to this point? Okay. So I studied fungus and I studied Drosophila, fruit flies. When I applied to PhD programs, I was most interested in studying human disease, and I was interested in the genetic basis of that disease. So when I was looking for a lab to join in my PhD program, I was focusing on studying humans, studying human disease, and specifically, I was interested in cognitive conditions, neurodevelopmental disorders, or congenital disorders that involve the brain. And so I was looking at labs where that was the focus. It was just what interested me most. And so in Don's lab, he had a project ongoing studying the genetic basis of autism. And so I sometimes describe my thesis work as throwing the genetic kitchen sink at autism. And so what we had were samples from 3 different regions of the brain. And so these were post-mortem samples. So individuals who had donated their bodies to science, their brains were frozen and sections from different parts of their brain were stored. And then different researchers would apply and get access to these samples. And so I had 3 different regions of the brain, and I had the same individuals, samples from the same individuals from these three different regions. So one was BA10, which is focused on executive function, BA44, 45 on semantic decision, and BA19 on visual association. And so the plan when I started my PhD was to study this at both the DNA and RNA level. So quick genetics 101, for those of you who haven't had a basic biology course for a while or haven't taken a genetics course, DNA is what's in every single one of your cells. You get 1 copy from maternal line, 1 copy from paternal line. And in each of your cells, you've got this double-stranded helical structure, and that's what gets passed on from generation to generation. RNA, on the other hand, is what gets made from DNA and goes on to make proteins, and proteins are what like makes your body function. And there's only 2 copies of DNA in each cell, but there's 0 to thousands and tens of thousands of RNA in each cell. So RNA is what allows your brain cells to be different from your heart cells, despite the DNA being the same. Comment, cells like the squares in my Jupyter notebook. Not quite. And that's the joke where, oh, there's so many words. Oh, there's one like coding, right? Like I teach coding and programming, but we code experiments. So if you take a video of a participant in a study and you look to see what happens throughout the experiment, you're coding, but it has nothing to do with programming. So there's all of these words that are used in same exact word used in 2 different ways throughout the like span of what I do. Okay, so we were going to from each of epigenetics, about to talk about that in just a second. And so we have DNA and RNA from each region of the brain. We would extract the DNA and the RNA. From DNA, we would get what's known as genotype. So that would tell me at positions throughout your genome, each of the individuals in the study, did they have a G, C, T, or A, right? Those are the 4 bases in DNA. So we had genotype level data. We also had methylation data. So if you've heard of epigenetics, if you're familiar with Iran Mukamel's research in our department, he studies methylation differences. And so this lets us know about regulation, what genes are turned on, what genes are turned off. We also had RNA. So this is if I looked at all of the RNAs in my sample, and so there's about 20,000 different RNAs, give or take, in a human genome. What is the level at each of those genes across all of the individuals in my study? And so these were the data that we were collecting. So I was doing the experiments to collect these data, and then had data across about 89 different individuals, and was doing the analysis at each of these. So which genes were differentially expressed between our control brains and our individuals affected with autism. Same thing, methylation between control brains and autism, and then genotype, looking for differences at the DNA level between controls and individuals affected with autism. And so after that, it was the relationship between these, studying how methylation affects gene expression, how genotype affects gene expression, those are called eQTLs, and then how genotype affects methylation, those are meQTLs. So each of these white sections at the bottom and lines between them is a different analysis to do. And the idea is to do that across all of the brain regions in our data set. So like this is what I started with as a graduate student. And the ultimate goal is to like understand the biological pathways and to know which genetic variants affect this condition. All right, so my main focus was at the level of RNA-seq, or doing RNA and understanding gene expression. And towards the end of my PhD, I studied the effect of methylation. So did not get to all of this in my PhD, but started chipping away at parts of it. Okay. So a lot of words on this slide. Details don't matter. What I want you to know is that all the blue started with RNA-seq read data going down to RNA genotypes, and then going over and down to the bottom are all decision points that have to be made when you are analyzing RNA-seq data. And that means there's a whole bunch of different softwares that you could use to do each of these steps. And I became frustrated as a PhD student. I was like, well, how do I know which one is the right one to use? Like, if I use Bowtie, which was one of the software, and don't use any of the others, how do I know that that's more right than any of the others? And so we were on a call where we were discussing these data, and I turned to my boss and I was like, well, can't I use EQTLs that we already know exist to figure out which decision is best? And I think that was like the best idea I've had of graduate school. Everything else in my PhD was very influenced by my advisor, by what we were already doing with what was going on in the lab. But that was one point where I was like, I can say for sure that was my idea. And I then was able to run with it. My boss liked it. And so I started using a bunch of different softwares, comparing them, and using EQTLs that we already know exist as gold standards, and said, okay, if I use software A, does it detect more of the EQTLs than if I use software B, C, or D? And so we use this to help guide how we were going to analyze our RNA-seq data. And again, details don't matter, but the proportion is on the y-axis of how many of the EQTLs that we know exist we detected along our analytical pipeline. And so we could see that as we removed outliers and used what's known as CQN for normalization and included covariates and included the outlier removal after that, we found more EQTLs so that we knew we were on the right path. So that was one of my papers from graduate school. Another one with a postdoc named Simone Gupta. We analyzed the differences once we knew what pipeline to use. We analyzed the differences between individuals with autism and the control brains that we had. What's on the y-axis in this top image are the chromosomes in a human. And the x-axis are our chromosomes. And the y-axis is the negative log 10 of the p-value. So each point is a different gene. And what we're measuring the p-value of is the difference between individuals with autism and unaffected controls at each gene. And so 2 of these genes are above this dotted line. This dotted line is genome-wide significance, meaning given all of the tests we did here, for each of these genes, these 2 we're most confident are actually different between controls and cases. And those might be what we would be interested in understanding the pathway of as we continue on to do further experiments. So those were the results of our RNA-seq analysis. Not getting into the details, we also analyzed these at the level of what's known as modules, so genes that are regulated similarly. I'm not going to walk through each of these, but I have that slide here so that I could say that we did that. So not just looking at single genes, but a whole bunch of genes that work together to understand pathways. And towards the end of my PhD, I looked at the effects of this methylation, this regulation on autism-affected brains relative to controls. The details, again, not super important, but CpG methylation is what's most heavily studied. What that means is there is a methyl group, a CH3, if you're coming from chemistry, added to the cytosine right next to a guanine. So the C is next to the G. CpH indicates the C is next to any of the other bases. And we found in autism, there was this increase in CpH methylation, in hypermethylation, meaning a lot more methylation in the context when C's are next to anything other than G's. And so ran a bunch of simulations and permutations to come up with the results that you see here. Again, not walking through the details here. Okay, that's a whirlwind through what I did in my PhD. And it's something I haven't talked about in years, but I was basically trying to understand the genetic basis of autism. So any questions up to that point? Okay, one of the best lessons I got from Don, my PhD advisor, was if it looks like a unicorn, it's probably a unicorn. It doesn't exist. And so one of Don's guiding wisdoms is that if you are doing a data analysis and it looks too good to be true, it might be too good to be true, but it is your job to convince yourself when doing data analysis that it is actually true. So you have to poke as many holes in it as possible to convince yourself that what you think is there is actually there. Too often in science, when we get a result that we're excited about, we stop and don't look harder. And it's a result of what Don refers to as a unicorn. Got too excited, made a mistake, didn't look hard enough at our data. And so it was Don who really instilled in me how important data analysis is and how long and hard we have to look at our data and how many holes we have to poke in it to convince ourselves what we're seeing is real. Don't get excited because you saw something you wanted to see. If you see something you're excited about, do everything you can to convince yourself that it's true. Okay. At the end of my PhD, I wasn't quite sure what my next path would be. I knew I would do a postdoc, but I didn't know from there what exactly I wanted. And so I got my PhD and I applied to a handful of different postdocs. I knew I wanted to pivot, happy to answer questions why I knew I didn't want to study the genetic basis of autism for the rest of my life. But I had also been following the microbiome field. So for those of you who don't know the microbiome field, it's studying the bacteria throughout our bodies. And a lot of these bacteria are good. We're covered in bacteria. I became really interested in this as an undergraduate and had just been reading papers for years and years and years, but I had no background in the microbiome field. So I applied to a bunch of microbiome labs. I also was asked by Jeff Leake, who ended up being my postdoc advisor, to do a postdoc with him. So he was on my thesis committee. He is a biostatistician at Hopkins, and he had sat through my meetings for years and years during my PhD. And towards the end, he said, "What are you doing? You should come do a postdoc with me." And I told him, "No, I shouldn't. That's a terrible idea. I'm not a statistician. And I don't have the math chops that you would need for me to be a postdoc with you." And he was like, "You're a great analyst. Forget about everything you just said. Come talk to me. We'll discuss possible paths forward." There was a comment in the chat. So if your code compiles the first time, you should be worried that something is wrong. I would say that's true unless your code is well tested. So if you've got a whole bunch of unit tests written and you've already done the work up front, if your code compiles, that's great because you've already run the tests. But yeah, too good to be true might mean that something's going wrong if you haven't dug in deeper. I like that analogy. Okay. So I got rejected from a whole bunch of microbiome labs. I had been told when I went to grad school that what you do in your PhD doesn't matter if you want to go become a professor, that what you do in your postdoc matters the most because that's what you take with you when you go become a professor. And in my mind, I was like, professor is what I think most, but I'm not totally decided. That was not true. When I applied to the microbiome field, it was a really hot field. Lots of people wanted to work in it. So of course, professors were hiring postdocs who had a background in the microbiome rather than me, even though I knew a lot about the field and was a good analyst. They were like, "Oh, of course I'm going to take this other person who already knows who has already done this work." And so I did ultimately get an offer to do a postdoc with a really great scientist named Ron Bleckman. And so I was deciding again between two things. It was either microbiome genetics research with Ron Bleckman or work with Jeff Leake and do a lot of genomic analysis, but not really focused on human disease. And so the reason, again, I chose to work with Jeff is because I was not set on working in the microbiome field in genetics. I figured Jeff would open up more doors. He's really famous in the biostats world, and he's awesome. And I knew he would be a good mentor. And so I decided ultimately to stay in Baltimore, where I already was, where I loved my life and had a really great community, working on stuff that sounded interesting to me, but knowing that when I did that, I was not going to be going down the microbiome path. Okay, so unfortunately, his name is spelled Jeff, not Geoff. It's Jeff. So similar into Jeff, who's here right now. And so Jeff Leake was my postdoc advisor. If you talk to enough people, you will hear bad stories about advisors from PhDs and postdocs. I had incredible mentors all along the way. So all four of these people are still people I talk to this day. These are still people I could ask questions about my career at any point in time, and they were incredible mentors. So one is I'm lucky they were great. The other is I prioritized that. I knew these horror stories. So when I got to my PhD, I talked to people in the lab and got a sense for what they all had to say about their advisor and how he mentors them and how they work together. and made my decision based on that. So some people pick a lab based on the most famous scientist, and that might be the right decision for them, but I knew I needed a lot of mentoring. I was coming straight out of undergrad. I knew I needed to learn a lot. And so I needed really strong mentors. And so part of it was intentional and part of it, I got lucky. These were just great people. Okay. How are we doing on time? I'm going to pause for questions and then go through this project quickly as well. Okay. So when I joined Jeff's lab, there was a lot of human data sitting on computers. So what you need to know about the field of biology is most work is grant funded. And so the government gives labs money to do their research. At the same time, big grant agencies said, if you want this money, at the end of your study, you need to make your data available. And so biologists for the past 10 years have been just depositing their huge data sets onto the internet where they just kind of sit. They're not easily usable. They're not easy to get out. And so what Jeff's group did was they started this project called Recount2, where we took all of the publicly available human RNA sequencing samples. So if you remember from my PhD, I did a lot of RNA-seq analysis. So I was very comfortable with these data, but now I was working at a scale beyond what I had worked at before. So I had 90 brains as a PhD student, and now I had 70,000 human samples. We took all of the publicly available human RNA sequencing data from the internet, processed it on a single analytical pipeline, made it all available to anybody who wanted to use it so that people could take those data that are already sitting there where people have already paid to do this and then answer their question of interest. So this had lots of controls, tons of different human conditions, diseases included in it. 3 main data sets are listed here at the bottom. That's where we got the 70,000 samples from. The problem was information about those samples was missing. So we had all these 70,000 samples, but we didn't have information about the phenotype, like whether or not what sex the individual was that it came from, whether or not, which tissue it came from. And so what I started to do in my postdoc was build a model that predicts these phenotypes from the expression data. So this was a more machine learning centric project than what I had done previously. And the idea was we had good phenotype information in 2 of the studies, GTEx and TCGA, and the SRA I refer to as the Wild West. There was tons of missingness, not helpful information. So the idea was I would train the model in half of the GTEx data. I would test it on the other half. I would then predict, see how it's doing in my validation data set in TCGA. And then ultimately, I would be able to predict phenotypes across the SRA samples. And so I did this by building an R package called PhenoPredict. I'm going to whirlwind click through this because the details aren't important here, but just to know the functions in the package, the main functions are listed here. What this did is it took expression data as input, determined which differed between my phenotype of interest. Here it was sex that I was predicting, found those regions that were of interest that could discriminate between my groups. I'm going to skip over all the math. Then extracted the coefficients measuring the relationship between expression and phenotype across all of those regions. Again, skipping the math. All these slides are available on my website. So if you want to look through them later. And then from this model that I built across all of these filtered regions, determine the likelihood of the phenotype coming from for each individual. And so for each person, summarized across all of the filtered regions and came up with this likelihood. So in this first column, this individual was most likely to be male. So I maximized this such that the first person, if I'm comparing 0.99 to 0.01, I would predict they would be male. Did that throughout the entire table. Got my predictions in my training data set, compared against my reported, got my prediction accuracy, then extracted those same regions in my new data set. So I built my model in the training data set, extracted those same regions, and then applied the estimates from my model to the extracted data. Again, skipping math, and made the predictions in this new data set. There was a comment, I wish it was possible to skip over the math more often in cogs. I think the math here is actually pretty cool, and the matrices here. So it's it's simple and it's straightforward, even though it looks more complicated than it is. So apologies, we're skipping it for all that who love math. Okay, so great question. What coding experience did I have before working on this model here? I did not write my first line of code until my PhD. So my first year of my PhD, I first was working on a project that I didn't talk about here, where I was trying to actually reproduce software that already existed so I could learn how to program. And so my advisor, Don, in my PhD, he programmed in Perl. No reason anybody here should have ever programmed in Perl. The postdoc programmed in Python. So that was the second time. I knew what Python was by that point. However, our postdoc was 8 months pregnant. And so she was going to be going on maternity leave soon. And so I was like, okay, well, if I have questions, I can't ask Simone because she's going to be with a newborn. And so I'll ask Don. And so I should learn Perl, which was a terrible mistake. I should have learned Python. So I learned Perl my first year of graduate school. From there, I mostly did programming in R. And then I continued to use and teach with R through my time at Hopkins. And so I had a lot of programming experience here, but it was all self-taught. And I wrote some really bad code throughout my PhD. Similar question, what math, stats, ML experience did I have leading up to this? Okay, a lot of this is, so these are the other things that you wouldn't know if I were in class because I don't tell students. My math background was I had Calc 1, Calc 2, I had 2 stats and probs courses. These are like the actual courses that I took. And then I picked a PhD program specifically that was my undergrad. So not a lot of math in undergrad. In my PhD, I picked a program specifically that required at least 2 stats courses. So this is grad level stats. So I took 2 grad level stats courses and then nothing. So any linear algebra that I needed to use here was math I taught myself. Any ML that I learned was ML I taught myself / asked my advisors to make sure I was learning correctly. So my formal programming and my formal math background are way less than what most of you have coming out of undergraduate. Yeah, so everyone who has a class with me is already one step ahead, which I try to say on the last day of COGS 18. Like they're certainly more formally trained than I was. All right, so the take homes from this, with skipping over all the details, is that we were able to accurately predict tissue. We were able to accurately predict sex. We were accurately able to predict whether it's a cell line or not a cell line. And that when tissue were mispredicted, it was to a biologically similar tissue. All right, I know we're getting close on time. I'll do this quickly. Halfway through my postdoc, so my postdoc was going to be 2 years. And so a year in, I went into Jeff's office and he had been bugging me like, what do you want to do after this postdoc? What do you want to do? How can I help you get there? And I went in there and I was like, Jeff, I want to teach. And he was like, great, you should probably get some teaching experience. And I said, okay, I've applied to this summer teaching job. If I get it, can I leave for 3 weeks? And he said, yeah, absolutely. And then I did that, loved it. Most I've ever worked in my life. But I really enjoyed it. When I got back, I continued to teach at Hopkins and started applying to more teaching-centric jobs. During that time while I was applying to jobs, I also built this data science education program online. Because we're short on time, I'm going to skip a bunch of things where February 2018 is when we started building this online program. And the goal was to just get people to learn the basics of data science. We met with a partner organization in April. We launched a month after. So this was like how to build an entire curriculum in 3 months. There was some software that helped make that easier that we also developed. And the courses in what was previously known as Chromebook Data Science, but is now known as cloud-based data science, are listed here. And so I built the content for most of these courses in the matter of months. It was a huge sprint. So we introduced how to use a Chromebook, taught the basics of data science and version control, introduced how to do analysis in R, and then focused on what are unfortunately often called soft skills. And all of that was part of what I did at the end of my postdoc. Oh my goodness, somebody said they took this course. Okay, it's still, this was a first iteration. It's free, anybody can take it. And we also built this in-person program where we taught this with in-person tutoring, gave free laptops, paid people for completion. and helped them with job search after it, and built that program in Baltimore before I left. So the impetus for building all the online content was like, oh, let's make it available to the world. But this was our main focus. How do we take individuals who had dropped out of high school but gotten their GED into a role where they would make a living that they were not able to make otherwise? Okay, so students started in May 21st for our first cohort. They were projected to finish in August. We had checkpoints. At each checkpoint, they got money for completing the courses. It, of course, took longer, as all things do when you start something new. Some of this was we were behind on developing, and students were waiting for us to get it to them. So it was not their fault. We launched CBDS on September 30th. We had students prep after finishing the first in-person session and present at the board meeting. And that was all in October. And then October 19th, I moved from Baltimore and arrived at UC San Diego to start on November 1. So it was a huge sprint as I was finishing up my postdoc. And so when I interviewed at UCSD, I did not talk about that program at all. But I had a bunch of teaching experience at that point. I knew I wanted to be into education. I applied to small liberal arts colleges, state schools, mid-sized education startups, local government data science positions, and a random editor position at a journal. And that's how I ended up here at UC San Diego. Could talk more details. I talk about some of them in COGS 108. These days, teaching, as you could imagine, is my main focus. The courses I tend to teach or have taught up to this point are here, and the quarters in which I taught them, with the number of students I've taught since winter quarter 2019. So it doesn't leave a ton of time for research, but the research I do mainly focuses now on what students do in my courses, how far is that off from what I was hoping they would get out of it, and how to improve from there. Learning how and what students learn, that's what I'm really interested in, but it's hard to study, right? I know how you do on tests. I know how you do on quizzes and assignments, but like, what do you really learn? And studying that is what I'm going to be working on going forward. Also, I have a collaboration at UC San Diego where we've been studying how THC affects driving and what the legal limit should be and how that should be measured. And so that's been really interesting work that I would, if it's 6 months from now, I'll be able to talk about it more. We just submitted those papers, so I can't talk about them publicly yet, but I will be able to soon. Okay, we're at 5. That was my whirlwind tour. What questions do y'all have? All right, there's a question in the chat. You've heard this term postdoc a lot since coming to UCSD, but like, what is different between a PhD and a postdoc? Okay, so to do a postdoc, you need a PhD. So first we need to talk about what a PhD is. A PhD involves coursework, typically for the first year or two, depending on the program. And then you do a deep dive on a topic of you and your mentors choosing for another 3 to 4 years. So a PhD typically, if you don't do a master's first, is like 5 to 7 years. And it's the coursework plus this deep dive. And you're part of a cohort, you come in with other graduate students, you have to do thesis committee meetings. And so there's structure around what you're doing. A postdoc is like a PhD in that you do a deep dive and you're doing research, you're working on a project or a few projects that are all your own, but you don't have any courses, there are no checkpoints, there's really no structure. So it's a very independent time, but you're also not a professor. So you don't have meetings, people don't really know that you're there all the time. So you have a ton of freedom to just kind of like delve into your work. So it's this nice transition time between PhD, very structured, have to do coursework, have to check boxes, and your professorship work, it's very busy, lots of email, lots of meetings, lots of things that don't feel like that your job, but you have to do. Where in a postdoc, you really just kind of like deep dive on your work. That's a really great question. Do you have any advice on preparing for technical job interviews in data science? Yeah, so I did a handful of them when I was interviewing around for the different jobs. My general advice would, if you haven't taken a SQL course or you don't know SQL yet, spend some time learning it because it's required of more than 70% of jobs that have data science in the title. And if you know Python and you know how to work with data, it will not take you that long to learn just the basics of SQL enough that you could honestly put it on your resume and you would be able to to use it. So you don't need to be a SQL master, but you would want that on your resume. So that would be first piece of advice. Second is when preparing specifically for the job, the technical aspect of the interviews, data scientists, more than computer science, software engineering jobs, will tend to give you a take home where they give you some data, they give you some task, and you have to prepare and do an analysis. So the more things you can do like that, the more prepared you will be. So doing side projects, I hate that because I know y'all are super busy and like who has time to do tons of side projects, but if you can fit them in, they don't have to be super complex, but then you can add them to your personal website and they help you prepare for interviews. The live coding interview portion, hopefully we're moving more and more away from that. If that happens, LeetCode is probably your best bet for how to prepare for that. The other one, it seems that most entry jobs need 2 plus 3 years experience, or they want a PhD. So one thing to do is, yes, that happens, but if you meet 70% of the requirements other than that, you might still want to apply. Now, if it says like PhD required, you might not, that might not be the best use of your time, but if it says like PhD preferred or 2 plus 3 years experience, if you can demonstrate that you have those years of experience in school, right, that you've done projects for 3 years, you can explain that in your cover letter. Advice for someone who has no idea what you want to do after undergrad. So if you have no idea, think back through the courses you took that you enjoy that piqued your interest, and then start going down that path. If you're having trouble deciding between a few, start looking up on LinkedIn and on Twitter people who have jobs similar to what you think you want, and try and get a sense for what they actually do. If you're curious, on LinkedIn, you can message people. Make sure you personalize it so that they don't think you're just spamming everybody, but say like, hey, I saw this about your background and your path. Can you tell me more about X, you know, the thing you're interested in? And that might be a way to gather more information. My other advice is, if you're not sure what to do about after undergrad, you should never default to a PhD. A PhD is a lot of work, and you don't make the money you would make if you had a job. And I've seen people do it and they dislike it. Now, a master's is a happy medium. If you want to learn more, but you don't want to dedicate 5 to 7 years of your life, that's something to consider. But that's another piece of advice, is like, you should only get a PhD if you really want a PhD. Yeah, have a few live coding interviews that are coming up. HackerRank, yeah, LeetCode will help you get ready for them. Any advice if I should get a job and try working first or apply for grad schools? No. The advice I'll give you is, it is certainly a personal decision. Some people have very strong feelings that you should go work before going to grad school and get experience. I had no problem going straight from undergrad to my PhD. I like school. I didn't feel I needed a break from school. So if you feel you need that break, go get a job and decide later if you want to go back to grad school. If you feel unprepared for the job force and monetarily, like going to grad school works for you and you want to learn more, apply to grad schools. But like, it's a very personal decision and there's not one right or wrong answer. Question: how much harder is it for you to conduct your own research ideas and hypotheses in a master's versus a PhD? It depends. So if you want to run your own lab at a university, it'll require a PhD almost for sure. If you want to do research at a company, there's a lot of R&D going on at many, many companies, and they hire masters a lot. You can then raise through the ranks and get more senior positions, regardless of whether you have a PhD, in many companies. In a company, you have less freedom on what you get to do with your research, because your research has to fit the ends of the company, right? Because they need to make money. But if the company is a good fit for you, it might be great to have a master's. You can certainly do master's level research at an institution, but you'll have less independence. Yeah, professorships, even ones centered almost entirely around teaching, require a PhD, correct? Yes, that's true. I have not seen one that does not require a PhD in a really long time. And part of this is because there are way too many PhDs and not enough professor positions. And so they do typically end up being very, very competitive. And so one way they can make it so that there aren't as many applicants is by requiring a PhD. There could be master's people with masters who would be great. But because they're so competitive, the PhD, most universities will require it by rule. And second is there's already too many applicants. So they won't need to lower the PhD requirement when they already are so competitive. Question, should we be worried if we don't have a job offer by the time we graduate? So less than 2 months left, got a bunch of HackerRank challenges and rejections and only 3 second round interviews so far. So starting to feel more nervous. So it is totally normal to feel nervous in that situation, but know that you aren't alone. A lot of people will be in that situation. It's a busy time and it's a competitive time. I've seen it work out at the wire for so many students. But know that I've also seen it that students wind up working a job that's not their ideal job for 6 months to a year, continuing to apply, and then getting into a job they're really excited about. And that's okay. Like all of these paths happen and they happen to very qualified people. So keep trying, but you shouldn't be panicking at all. Even if you end up in a job that's not exactly what you wanted, you can find your way. You saw my path. It didn't make any linear sense at all. You can find your way to a really a job that fits you well. Laura, I see your hand. Go ahead. What's up?
Hi, Prof. Ellis. Thank you so much for sharing your story with us. I took COGS 18 and I was just wondering like what other classes you're going to be teaching next fall or next winter.
Yeah, I'm going to pull up my little note that I have that tells me which ones I'm teaching when so that I don't tell you the wrong information. Okay, so I have a different schedule next year than what I've had historically. So in the fall, I'm teaching COGS 18 and I'm teaching COGS 137. That's a new course. And that is, I don't remember the name of it, even though I made the name. So it's a course that I proposed and it's going to be focused on data analysis in R. So it's going to be like a hybrid between 18 and 108 in that the first third of this R course is going to be R syntax. So there will be no programming prereq. But then there's going to be a second part and a third part that are going to deep dive into like the statistical analysis of specific approaches and specific analyses. So COGS 108 is very broad. This COGS 137 is going to be much deeper, but on specific like statistical analyses that we just don't have time to get into in 108. So that's the fall. In the winter quarter next year, I'm slated to teach COGS 108 and COGS 169. 169 is a genetics course in cognitive science. And in the spring next quarter, I'm only slated to teach COGS 9. So that's intro to data science.
Do you recommend taking COGS 108 before 137?
I think order doesn't matter there.
Okay. Okay, good to know.
Yeah.
Thank you so much.
No problem. Question in the chat. This is a good question. Why did I go PhD route rather than MD PhD? I'll answer that question first. So the MD PhD offer I got was late after I was on the wait list. So it was at the University of Iowa. So for background, I only interviewed at 2 MD PhD programs. One was the Mayo Clinic and one was the University of Iowa. I did not get a good feeling at the Mayo Clinic. It was not going to be a good fit for a number of different reasons. And so I pulled my application after the interview. I was like, you know what? So I don't even know if I would have gotten in there. I have a feeling I wouldn't have been. So then the only place I had interviewed other than the Mayo Clinic was University of Iowa. I was on their wait list for a while. In the meantime, all of my PhD offers came in. And I had decided at that point that I was going to get my PhD at Johns Hopkins. So by the time I got off the wait list at University of Iowa, I had decided that like the PhD at Hopkins was what I wanted, but it was somewhat of timing. I think had I gotten the University of Iowa earlier, I may have made a different decision. The second question here, would you recommend doing a master's if I'm not sure about doing a PhD or an MD yet? That's a tough one. So something to keep in mind in the biological sciences. So if you're going in any of those, a PhD does not cost money, but a master's does. And so something else you should know is, and that I did not know before graduate school, was that if you enroll in a PhD program and you do all the coursework and you pass your comprehensive exams and then decide to leave, you would leave with what's known as a terminal master's. So it would be a master's degree. And so I think if you're like close on the line between PhD and master's, I would probably try for the master's. Now, I don't think you should join a PhD program intending to leave after the comprehensive exams to get your master's for free. But if you're on that line, consider that. Now, if money is not an issue, any decision is fine. But for most students, money matters, as it did for me. And so you have to consider that in whether a master's is a good fit. One thing to keep in mind is if you're thinking you're going to go the MD route, a master's in public health can wind up being, so an MPH can wind up being a really great option that fits well with an MD. But yeah, it just kind of really matters as to what you ultimately want to do. Question. I'm really interested in what you were talking about earlier with how people learn better. Do you know any books, websites, or whatever that are good introduction to that kind of stuff? I've been talking for an hour now. I can't remember what I said. When was I talking about how people learn better? Oh, from my research, the like last thing that I was saying. That part? Oh, okay. So there are a few what are they, conferences, that's the word, that really have a lot of papers on this. So one would be SIGSI. I think that's the acronym. The other would be CHI, so the Computer Human Interaction Conference. And so if you were to look up the ACM website and papers on ACM, so I'm just throwing them in the chat, there's a lot to be read in this like realm on those sites. So if you were to like, if you are struggling to get the full paper that you wanted to read from there, know that if you go through the UCSD VPN, you can usually get the full paper if it's restricted otherwise. And so papers in that realm, but books wise, I should know more. I'm reading Thinking Fast and Slow right now, which is kind of on that topic, but it's pretty long and dense. So I wouldn't say start there. But I would say like looking into education papers, and that would be a good place to start. That would be my suggestion. If I think of anything else, I'll let Christina know and we can then share it elsewhere. Oh, thanks, Jeff, for sharing that in the chat. I'm going to put that on the video just in case anybody's listening to this later on. Psych professor recommended Make It Stick, The Science of Successful Learning by Brown, Roediger, and McDaniel. And hopefully I said that second person's last name correctly. Sorry I went over, y'all. I had intended to finish earlier, but I appreciate everybody asking questions after the fact. Great. Glad it was interesting. Feel free to ever email me if you have any questions. We can chat, follow-ups. And I hope everybody has a great rest of your day.
Yeah, thank you for coming and talking and answering everyone's questions.
Thank you for having me. Thanks, y'all, in the chat.
Cool. Okay. I'll stop the recording now.